This one is by Axovia Therapeutics, which just has recently been announced.
Hi, I'm Phil Beales. I'm CEO of Axovia Therapeutics. I'm also a clinician scientist at UCL, where my lab is based. Axovia is a new gene therapy company that is built on strong clinical and scientific foundations in the ciliopathies. So we're focused purely on ciliopathy disorders. Our early proof of concept work in our lead asset, which is Bardet-Biedl Syndrome, BBS for short, has demonstrated we can prevent early onset blindness and severe obesity using AAV gene therapy. So it's the focus on a broad group of collectively common diseases with unmet needs that we believe is our key differentiator. We've got early insights into the biological basis for these diseases, including involvement in primary gene discoveries, but most importantly, access to patients. From our vantage point, there are no competing therapies on the horizon, and we have IP protection that allows us full freedom to operate. And through our partnership with Jaguar Gene Therapy, we are guaranteed scalable vector manufacturing right the way through from preclinical studies to first-in-human trials. And we're utilizing a proven AAV platform that is supporting our proposed multiple routes of administration, which I believe will become much more commonplace as most rare diseases affect multiple organs. So Axovia is a spin-out from UCL, where Victor Hernandez and I developed the research that holds the promise of this new transformative gene therapy that could prevent blindness, obesity, and neurological sequelae in patients with BBS. Ciliopathies comprise a group of over 40 disorders and counting that have a common pathomechanism, and that is dysfunction of cilia. Cilia are present on virtually all cells in our body, and we now know that cilia play an important role in both organ development and tissue maintenance throughout life. This explains why virtually every organ in the body may manifest clinical defects depending on the mutated cilia gene and the disease in question. Some of these features are shown on the mannequin diagram on the right-hand side, and you can see the acronyms for several of those ciliopathy diseases. But our first indication is for Bardet-Biedl Syndrome, as I've mentioned, and the two genes in particular that we're focused on are BBS1 and BBS10, which together account for 70% of all patients with BBS. The development of these gene therapies is the culmination of over 2 decades' work in both the clinical and the scientific arena. 10 years ago, I was able to establish through the NHS a national clinical service for all BBS patients in the country to provide them with lifelong care delivered by a multidisciplinary team of 7 specialists. We also provide a national diagnostic service for the 22 genes associated with BBS, and we collate the data into a national BBS registry that contains over 600,000 data points, and it's forming the basis of our natural history review. In parallel, in the Ciliopathies lab at UCL over the past 20 years, we've discovered dozens of novel disease genes. We've defined the international diagnostic criteria for BBS, and we've contributed to our understanding of cilia biology and the role of cilia in disease pathogenesis. Around 7 years ago, we started our gene therapy program that led us to show the benefit of gene therapy in BBS1 murine models in terms of both prevention of retinal degeneration as well as the rescue of obesity and brain defects. These are the 2 routes of administration we're proposing for our gene therapy: an intracerebroventricular injection and a subretinal injection, as both these deliver products as close as possible to the target tissues in a one-time delivery. We know from case reports that BBS is found in almost all populations worldwide, albeit with differing prevalence rates. However, we can see just in the US and the EU alone, there are over 9,000 patients with Bardet-Biedl syndrome. Axovia is dedicated to accelerating a robust ciliopathy pipeline that is based on our scientific know-how and clinical experience. Although BBS-1 is our first asset, for which we are on target to deliver first-in-human trials by the end of 2022, this will be quickly followed by our second asset, BBS-10. Joubert syndrome is a predominantly severe neurological condition without any treatment availability, and it's currently in our discovery phase. It also has a slightly higher prevalence rate over BBS. How has the journey been so far? After we licensed our BBS-1 asset to Deerfield Management in early 2020, we went on to complete a Series A in partnership with UCLTF Fund later the same year. And this has allowed us to build the infrastructure for the company, including our lab based in the King's Cross area, close to the Crick Institute, UCL and GOSH, where we expect the first trial site to open at the end of next year. We've invested heavily in our manufacturing capability, developed a robust medical affairs program and strong regulatory framework. In fact, in October last year, we received orphan drug designation and rare pediatric disease designation for BBS from the FDA. We've also had endorsement for our BBS-1 program plan through early interactions with the FDA and the MHRA. And we're working actively to secure early patient engagement and education around gene therapy. The biotech sector is currently booming, and so we're excited to take this opportunity to be able to turn early stage research into transformative therapies for patients. Thank you very much for your attention.
Thanks, Phil, for the presentation, and I'm also really pleased to announce or welcome as well Victor Hernandez, who's co-founder and CSO of Axovia. Good evening to both of you. Really great to have you here on our showcase. And I guess, first of all, again, for the audience, please raise any questions you might have. But from my side as well, it's really great to hear your commitment to, I guess, the London area, where we also have a UCL and all our UCL partner hospitals, which together, I guess, with as well the UCL Technology Fund really can provide really the requirements or all capabilities to really bring something to the clinic. So I guess my first question really is about going a little bit back to the panel that we had earlier on about safety in gene therapies and so on, and quite interested to see in your presentation that you're suggesting for your initial vector 2 different delivery routes, and just wanted to hear your thoughts about that. How did it come to that? And as well, is there anything that you think might have negative effects, given that others really already have these questions where it's only one delivery route?
Okay, yeah, well, it's a pleasure to be here, Dominik. Thank you for inviting us. So the way in which we actually got to this point around particularly routes of delivery, routes of administration, really is related to the condition itself that we see in patients. And so patients go blind very early on. They start to go night blind at around 4 years of age, and that progresses very quickly over the course of the next decade. So that most patients are all blind by the time they get to the second decade. And that means that in terms of the route of administration, it makes sense. And because photoreceptors are the predominant degenerative cell, then a subretinal delivery is the obvious route of administration there. Our choice for the second route of administration was more fortuitous, should we say, because we weren't necessarily looking to adopt that particular approach. It really came from the fact that we knew that if we were going to affect obesity, for example, we knew that it's driven by hyperphagia. We know it's driven by a central process within the brain, the appetite center. So therefore, it made sense to deliver the therapy into the brain.
Great. And another question I have, obviously, over the last couple of months with Axovia, we really have heavily invested on manufacturing regulatory aspects. Victor, I was just wondering, looking back, is there anything that you wished you had approached or thought of before it was in a spin-out company? So maybe just as a hint for other academics here on the showcase to look out for at an early stage.
So I guess that for us, we're a special case because we were very lucky to have Jaguar Gene Therapy, our partner company, supporting us. And the Jaguar team is formed by the Avexit team, the team that moved Zolgensma into the market. So from the beginning, we had their support in terms of regulatory and CMC. So that means that perhaps we are not the best suited to tell everybody else about this process. We are extremely lucky to have them on board and to support and guide us in this pathway.
That's great. And given that we are coming towards the end of the showcase, so what's next for Axovia? What are your next immediate steps? Maybe something that's not directly in the presentation or something like that you just wanted to make people aware of what we should potentially be looking out for.
Well, I can say that we're at the point now where we have really built an incredibly strong structure. But what we need to do is build out the company and the capability and the capacity moving forward. And so we are working toward a Series B round, which will be probably opening this summer. And that will help us do three things. The first is to take the first BBS1 asset into the phase 1, first in human trials, which, as I say, are slated for the end of next year. It will also allow us to do BBS10, which is the next asset that's lined up. And the beauty of BBS10, it would be a plug and play. We've already done the natural history on the patients. We know where the patients are. They're all genotyped. So we would plug that into an AAV9 as well. And then the third part of any funding from that particular raise would be to promote our discovery genes as such moving forward in our other ciliopathy indications.
Perfect. That's really a great end to I guess the company presentations of the showcase. And I guess on your last points, given that you mentioned that there are around 40 different ciliopathies, I'm pretty sure you will be quite busy over the next couple of months and years, and hopefully your plug and play approach will really be working. So again, thank you, Victor and Phil, for your time and the audience. Next, we will have